Okay, so one area where data availability has really exploded in the last 10 years is genetics. And what I want to talk about today is one of the ways in which such genetic data could be used, namely to construct predictors of various outcomes that we care about in the social sciences. And then I'll try to say something about how such predictors could potentially be used in social science research on child development and touch upon other applications as well. So I want to just say something very briefly about the kind of data that we'll be using to construct these predictors. And the predictors, which are known as polygenic scores, because they aggregate many genetic markers into a single number. I'll describe what those polygenic scores are. I'll talk a little bit about potential uses and limitations, and then I'll conclude. So very briefly, human DNA is a sequence of roughly 3 billion pairs of nucleotides that are spread across 22 autosomes and a sex chromosome. This sequence has subsequences that are called genes. They basically provide instructions for building proteins. And the vast majority of these locations, of these 3 billion locations, there are no differences across people in the nucleotides that they're endowed with. But at some locations there are differences. And such locations are sometimes known as SNPs, so single nucleotide polymorphism. So single locations in this nucleotide sequence where it's known that people differ. And typically at one of these locations there are only 2 possible nucleotides that can occur, and one of them obviously is more common than the other, and we can call that one the major allele. And so your genotype is just going to, we can adopt the convention of calling your genotype the number of minor alleles. And so any one person either has 0 minor alleles, 1 minor allele, or 2 minor alleles. Those are the only possibilities. One of them was transmitted from your mother, one of them was transmitted from your biological father. Okay, so, but when I say genotype at a SNP, I just mean a number that describes the number of minor alleles you're endowed with at that SNP. Okay. So genetic data has a number of distinctive features. One of them is that they're predetermined at conception. That's going to turn out to be useful in certain settings. For example, if you collect a biological specimen from a 3-month-old infant and genotype the infant, you're going to get near identical results to the ones you would get if you collected it when this infant has turned into a 90-year-old. The genotypes are randomly assigned, conditional on parents, at least at conception. In principle, we can find out a lot about this biological function. And it's finite and fully measurable, and costs that are just falling dramatically. And this is really the main reason why I think public conversation, genetic data will only become more widely used in the social sciences in the year ahead. So there are 2 types of techniques for measuring DNA. One is called sequences. When you sequence somebody's DNA, you measure the exact sequence of nucleotides at some location, or in this case, in the whole genome, or sometimes just around genes. Genotyping, by contrast, means that you have some array that defines a number of SNP locations that you then try to measure, and the SNP locations are typically chosen from some reference sample that allows you to sort of identify SNPs, so that you know in some sense that these are going to be areas where there's common variation across individuals. And these notice the logarithmic axis here, so that the cost of sequences, I think the first sequenced human genome, took an international team of hundreds of scientists and 10 years of work, and now you can do it for around $1,000, and genotyping costs have fallen to something like $20, $30. So and by genotyping, I mean SNP genotyping. So you measure somebody's SNP genotypes at something like 2 million locations. And that gives you pretty good coverage of most of the common variation in somebody's genome. Okay, so simplifying somewhat, a polygenic score, so think about somebody's outcome as determined by a bunch of some weighted sum of SNPs, each of which has some effect. SNPs are indexed by K, individuals are indexed by I, and then you have some disturbance that includes all other factors that influence the outcome, including interactions between genes. So typically when people talk about polygenic scores, they have in mind like some index of additive effects. So this thing, which we're going to call like a SNP, is basically, so if we standardize our outcome here to have variance of 1, and then you take the variance on both sides, you get that you basically get two terms on the right-hand side, assuming these things are independent. So you get that 1 is equal to the variance of this thing, a SNP, and the variance of the disturbance. And the variance of this a SNP is usually what's called the SNP heritability, so the total fraction of variance that's accounted for by the genetic markers that you've measured. So this number may be smaller than the total fraction of variance accounted for by genes as a whole, but we have some k SNPs that we've measured, and we want to predict our outcome. So H2 SNP tells us the maximum prediction accuracy that we could get if we could construct this A SNP. So by first person's true polygenic score, I mean a weighted sum of his or her genotype at these k locations where each weight is given by the true population regression coefficient. Of course, the problem is we don't usually observe we don't observe these weights, and so we have to estimate them. And usually, what you do is you estimate them from some non-overlapping reference sample that can be very large. And what's happened in the last 3 or 4 years is that it's become possible to estimate these weights with a lot more precision. So it used to be the case that you were mostly, that most of the variation in these beta hats was just noise, estimation error. But now, as samples have increased from say 10,000 people to 1 million people, there's a lot more genetic signal in these beta hats. So think of the PGSs that you can actually construct as a kind of noisy measure of the true PGS. And a nice thing about polygenic scores is that they allow you to capture information about all the SNPs in the genome, not just those that individually show strong evidence of association with some outcome. So if you construct a polygenic score and just restrict it to SNPs that in some independent studies showed very strong evidence of association at a p-value of, say, less than 5 times 10 to the negative 8, and then implicitly weight all other SNPs, assign all other SNPs a weight of 0, your prediction accuracy is about 1%. This is just to illustrate with polygenic scores constructed to predict somebody's years of schooling. And then as you progressively make this p-value threshold more and more liberal, the prediction accuracy increases a fair amount to about 10% in this case. Okay, so if we have all these unbiased estimates, then you can think of the estimated effect of some SNP as composed of two terms, the true population regression coefficient and some mean zero estimation error. And basically what that boils down to is the fact that the SNP, the PGS that we can construct given available data can be written as a sum of the true PGS and some measurement error. And this measurement error, but really what's happening is that this measurement error for a lot of outcomes is now very, very small relative to A SNPs. So the variance of the A hat SNPs that we can now construct for some outcome is mostly accounted for by variance in the this A SNP variable. Okay, so now for the rest of the talk, I'll show you some results of the prediction accuracy of polygenic scores for educational attainment and possibly also some other outcomes. And so when I talk about the R square of PGS, I mean the R square from a regression of some outcome on a polygenic score. Now this number, we know it can't be bigger than H2 SNP. That's the maximum prediction accuracy you could get if you knew the true weights. And in the limit of an infinite sample, we would sort of approach asymptote towards it.
David,
yes.
To interrupt, but I'm going to try and think about the application of this to something like height,
Sure.
which is a little bit, I think, less emotive than thinking about somebody's educational attainment being determined by their genes. So for height, we know that there is a genetic component and there's also a big nutrition component. So if you have a population where people are not very well nourished,
Yes.
lack of nutrition is going to be the main determinant of their height.
Yes.
And then as you move to optimal the genetic factors would be the main determinants. So what that means in practice is that the betas are going to
Absolutely,
change.
yeah. Mm-hmm.
And so that's what I don't understand about this exercise.
No, so the betas, these betas are not should not be thought of as structural parameters. They should be interpreted as reduced form parameters. And actually, one of the ways you can use these polygenic scores is to study precisely the issue that you're asking about. So you could ask the question: how much variability is there in the true betas across populations, across environments, across cohorts? And so there are all kinds of interpretational issues that arise when you do these sorts of analyses, I don't really.
But I feel like in maybe not in your applications, but in a lot of applications, those subtleties are lost. So you get somebody like throwing this in, oh, this is a predictor.
Oh, I agree with you. Yes.
This is a predictor of somebody's educational attainment based on their genes.
Yeah. Okay. So in the height example, there are a couple of important caveats. I was going to get to them later, but we can talk about it now. In the height example, the first thing is that yes, these betas may, in fact, they probably do vary a lot depending on the overall nutritional quality. The second point is that the betas could be a specific SNP could be influencing your height through some channel that we think of as environmental. And maybe BMI is a better example. So suppose that there are certain genes that increase the likelihood that you consume certain foods, and that's the mechanism through which the genetic variant ultimately influences your BMI. In common parlance, we would probably think of that as something environmental. Ultimately, it's what I put into my mouth, what I consume, what I choose to eat. But the pathway goes from the gene. But obviously, that's a pathway that one could interfere with or intervene with, and there's no presumption here that the genetic variant is influencing the outcome ultimately through some biologically proximal mechanism. And then the third thing I'd say is that in practice, there are always reasons to be concerned that in the original estimation stage, you haven't completely controlled for all the potential confounds that one may want to worry about. So maybe a certain allele is more likely to be found in certain environments, and if you detect an association with a gene, that's partly what you're picking up in the discovery stage, and that's partly why you get some of this predictive power.
Mm-hmm. That's almost certain to be the case in a GWAS study.
Yes, and in fact, and in fact, no, in fact, and we can quantify how big that that bias is, and it's actually one of the most important, I think, conclusions in our most recent paper. So I'm happy to talk more about that. Yeah. Okay. So yeah, I think it's important to think of these variables as predictors and not to overinterpret what they mean. Okay.
That seems like a good source of hypotheses. I mean, so kind of once you've done it, you could maybe say, look, in this population it was this level, this population it was that level. Maybe it went through, maybe changed with cheese consumption. But I don't see how, it's certainly science to be uncovering these relations if it turns out that gets you to be thinking about the channel. That's all positive, I think. I find it so. So I would go to
you might want to be measuring different betas for different populations, though. That's, I think, part of the issue.
But certainly, but then you'd have to measure them for one and then do it for another. I mean, I just don't see, that's what you're going to do. Like, it was different with an attentional intervention. The effect of a gene that goes through attention is going to be much smaller with enzyme. That would be the way I would interpret it. You find a channel, the channel is interesting, and it may be something that you could play with after that event.
Okay, so
David, this may be an interesting technical detail, but it seems to me the big thing you want to understand is in an additive separability sense, how can these genes predict something of interest? But then the question is what's the thing of interest just in the functional form sense? So it's a level of log. I mean, there's a little bit of optimal transformations of dependent and independent variables that goes back almost 55 years.
Sure.
What I would take is I would say this is a thing called an educational measure. I've had a set of regressors. I want to ask what transformation of the educational measure and what associated transformation of the individual controls give me the best goodness of fit. Is that everybody's?
No, that's true, and I think it's especially an issue where your outcome is something ordinal, like a response scale where you say very little to a lot, and then you have to assign some numerical value to each of these responses for any sort of practical application.
It does strike me as interesting. I mean, what Steve's saying, and like I don't know, but it does strike me as interesting because these aren't naturally, the numbers aren't.
No, that's true. Yeah,
not natural in any real sense.
Fair enough. Yeah. Okay, so it turns out that under some conditions, and I have like bonus slides on this if people are interested in the technical details, the R square of this PGS, the rate at which it's going to increase, you can actually work it out analytically. It depends on the overall SNP heritability, so the total proportion of variance explained by these genetic markers that you have available, population genetic parameter called K that we can quantify from other sources, and then N, the sample size. And it turns out that if you plot this function, we'll use for plausible parameter values and then have R squared PGS on the y-axis and sample size on the n-axis, what you see is that there's dramatic growth up until about 1 million individuals. So the ability, the prediction accuracy of these predictors should increase dramatically up to around 1 million. And after that, the gains are much more modest. After that, you're basically very close to the SNP-based heritability, and any additional gains have to come either from adding rarer variants or modeling interactions between SNPs or something like that. And this is actually what you see. So here we're just showing again from our own work how a sample size is related to the actual prediction accuracy. And the polygenic scores here are constructed using identical methods in identical samples. So everything is held constant except the size of the discovery sample. And it turns out that these numbers match the date-weighted predictions quite well. But consistent with the point that was made earlier, the rate of the sort of level is systematically a little bit lower than you might than we probably expected originally. I think we were probably expecting to be at about 15-16% by now. We're at 11-12%, and the most plausible is probably the case. This has a number of explanations, but it's quite plausible that one of them is just that these betas aren't exactly the same across cohorts, and the theoretical calculation makes this simplifying assumption that the correlation of the betas across discovery and validations and the training and validation sample is 1. In reality, it's probably something closer to 0.8. Okay, and it turns out that these predictors, based on the most recent, on the largest available sample size, also have some reasonable predictive power for all kinds of measures of scholastic achievement, years of schooling, probability of grade retention, these sorts of things. Now, to put them into perspective, here I'm just showing you that it's still the case that if you want to predict somebody's years of schooling, you're better off knowing the child's mother's or father's years of schooling, or better still both. But the early years PGS that we can currently construct is sort of on par, does a little bit better than just household income. Now, again, I emphasize these are just statistical relationships. One shouldn't overinterpret them. And the other thing is that here, and also these are all from. These numbers are not the incremental values of the PGS conditional on already knowing the parental education level. So if you already know the parental education levels and throw in something like a PGS, you maybe add 3-4 percentage points. So the prediction accuracy, like the incremental prediction accuracy, obviously depends on what's already available. Okay, so now I want to say something about how this could potentially be used. The PGSs are already being used in a number of ways. One of them is you could study treatment effect heterogeneity by genotype. My favorite paper on this was just published in PNAS by Barcelos et al. It combines in a very nice way a UK quasi-experiment that raised the compulsory schooling age and then polygenic scores. And it finds that the effect, the betas of this polygenic score changed as a result of the school reform. And just recently on bioRxiv, I saw a paper claiming that that if you construct polygenic scores for depression and depressive symptoms in a sample of patients with bipolar, you can actually predict how they will respond to lithium treatment based on this polygenic score. A second potential use of these scores is to identify at-risk individuals. And this is still very speculative, and I think there are a lot of claims being made about this sort of applications that aren't really backed by the by what's currently scientifically possible. But in principle, you can imagine that there are settings in which genetic information could be useful. And my favorite example of this actually comes from two people I know. They're siblings. They're now in their 40s, but growing up, they both had quite serious hearing impairment. And the older sibling, the brother, in his case, it wasn't diagnosed until he was 5 years old. And so his cognitive development was actually quite seriously delayed because of this. There was a lot of frustration. People didn't realize that the main reason he started speaking late and developed more slowly than his peers was that he just couldn't hear what people were saying to him. And in the case of hearing, I like that example because it's obvious what the remedy is. The remedy is if you could identify this kid before the child can speak, so the child has very limited ways of communicating the problem to his or her parents, but there are things the parents could do to remedy the issue: give the child hearing aids, articulate your words really clearly, make sure that the child looks at you when you're speaking, these sorts of things. And in the case of his sister, it turned out that she had exactly the same recessive condition, even though interestingly neither parent did. So there's no family history of this that you could have used to infer it. And in the case of the sister, they detected it really early, and she's mentioned to me on multiple occasions that she feels very, very lucky. And in this specific case, it actually could have been very high likelihood been detected by some genetic test. People have speculated about all kinds of other applications. One important distinction between the example I just gave and polygenic scores is that it's very unlikely that polygenic scores for very complex things like your educational attainment are ever going to come close in terms of prediction accuracy to tests for these kind of recessive genetic disorders. And the most important, I think, use of the polygenic scores is just going to be as a tool and as a control variable. And the reason, again, has to do with the interpretation. They're quite useful in terms of when they're used, or at least they will be quite useful soon, when they're used as prediction tools. When they're used in any other setting, you're back to this problem that Steve raised, namely that if you're making an argument that it's a credible instrument or that it can help you identify some causal effects, you really need to think about, like, substantively what you're assuming in order for that claim to be accurate. And there may be cases where that's appropriate, but it's not that these polygenic scores or genes per se are some sort of panacea. One always has to worry about exclusion, for example, in virtually every application that I've seen. So I think that in the short run, most of the useful applications would be to use these polygenic scores as tools in say, randomized controlled trials. And I'll have a little illustration of this. Now, there are two limitations I want to emphasize here. So my claim is that these polygenic scores, for outcomes that are widely measured already in existing data sets, such as height or years of schooling, we're already in the region of the parameter space where our genetic predictors are actually quite close to the SNP heritability. And for many other outcomes, we're getting there quite quickly. There is a problem, though, that if you train your score, if you estimate these weights in a sample of one ancestry composition and then use these beta hats to generate the predictor in a sample of a different ancestry, there's a lot of attenuation. And fundamentally, that's because the optimal weights may not be the same, and that could have two reasons. It could just be that the true betas are the same because of gene-environment interactions. And it could also be that the measured genotypes that we have are more strongly correlated with the causal variance in your discovery sample than they are in the replication sample. And there's very strong evidence that this contributes to the attenuation that one sees in practice. So if you take a sample of Europeans and estimate these beta hat Ks, and then you use those beta hat Ks to generate a predictor in the Korean population, say, there will be substantial attenuation. And the most obvious solution is to just generate larger data set of genotyped individuals from Korea, but we're not there yet. And in the meantime, applications to non-Europeans are are going to be generate more bang for the buck. Yeah, please.
So I'm just wondering, is the search for these phenotypes dependent on the outcome variable? Because then we're going to, say, a non-European population, and we're looking at, I don't know what the Korean outcome variable we're going to be looking for, test scores?
Oh.
Or,
So that could be another source of heterogeneity that causes attenuation. So that's certainly true. So, and I guess you could think of that as introducing heterogeneity in the true betas because you don't have the same outcome. So that's all.
and it may also be that the outcome could be culturally specific.
Absolutely,
And-
yeah,
okay.
yeah, yeah. No, there's no question. Yeah.
Other examples which are invariant across populations.
In every comparison I've seen of polygenic
Fine,
score,
Let me say
Oh.
interesting ones.
whether
You get the same answer for both for different populations. And obviously,
yeah, I don't know of any. Yeah, I was
you
going to
look at a
say.
paper that's suggestive of it, which would be nicotine. So if you have markers that tell you something about the response to nicotine, you would think that it's
Yes, it may be.
smoking regardless. But the fact that it isn't, that's extremely interesting.
Well, so that's true. So it's certainly the case. Basically, you get these problems. This kind of problem is limited to regions of the genome where the patterns of correlation with unobserved markers are different across different populations. Now, if you already know the causal variant, then it's much more straightforward to just check if the betas are the same. Yeah, and so there are for single SNPs, I think the answer is yes. For polygenic scores, I've never seen an example.
What were you asking? Polygenic scores or
It's
single
just
space?
paper. I'm asking about SNPs.
Yeah,
I want
for
cases where you know the biological pathway.
Yeah.
Oh, sure, all the time.
And so my question was, if you ran this regression, you get the same answer
No.
in different populations. If you don't, that's extremely powerful.
What he's looking at is a regression of basically up to 4 million.
No, I understand.
Yeah, but I think it's plausible that for a subset of these, you would get very similar results. And I think we know something about how to identify that subset of SNPs. Yeah. Okay. So the other thing is that the polygenic scores themselves are not very informative about mechanisms, not in and of themselves. It's hard to specify what they're captured. For things like education, it's almost certainly the case that partly they're capturing something about the environment in you were raised. There's no guarantee that they necessarily capture biological processes. This is what we talked about earlier with BMI and nutrition. So for example, one of the earliest findings was that there was a SNP in a gene called FTO that actually had a pretty substantial effect on body mass index in adults. I don't know what the latest evidence is on the mechanism, but at least in principle, you could imagine that this SNP is influencing BMI through some mechanism that varies across environments and that we would think of as environmental. So oftentimes, interpreting the polygenic scores as structural parameters or as instrumental variables is difficult to justify, which is not to say that there will never be applications that are credible. So these are important limitations, I think, and they're the reason why I think, in the short run at least, the most exciting applications are going to be ones where the polygenic score is just used as a control. And to illustrate, let me just follow up on a simple calculation we did back in a 2014 paper. So because we want some way to sort of quantify these gains in statistical power that you would get from having a polygenic score. So imagine a researcher who's trying to maximize statistical power to detect some treatment effect and has to choose between two options. Either you perform your randomized controlled trial in a sample of NX subjects, a proportion of whom are randomly assigned to a treatment, or you perform the study in a sample Q times NX experimental participants, a proportion of whom are randomly assigned to the treatment, but you get the polygenic score. Okay? And suppose that in both scenarios you have some baseline characteristics that explain 10% of outcome variance. So given X, what is the value of Q at which you would be indifferent between these two options? And of course that depends on the incremental predictive power of the polygenic score. But for a polygenic score with an incremental prediction accuracy of about 12%, you could reduce the sample size by preserving power by up to 13%. So is 13% a small number or a big number? Well, it depends on the kind of the costliness of the intervention. So that's the benefit in terms of the potential sample size reduction And the cost, of course, depends on what kind of intervention you're doing. But at least for, there are currently randomized controlled trials in the US underway in which they plan to give a group of treated individuals a substantial boost in their monthly income, basically to explore the possible effects of guaranteed basic income. In those sorts of studies, look, the cost per subject is of the order of tens of thousands of dollars, whereas the cost of gathering the polygenic score, which basically boils down to getting a saliva sample, is 1 to 2 orders of magnitude smaller. So it's already the case that there are applications where these polygenic scores are potentially useful as tools. Okay, so that's what I wanted to say about that. So to conclude, from a single DNA sample, I think it's already possible to construct polygenic scores for dozens of outcomes. And in the coming years, as the sample sizes that are used to estimate these beta hat weights increase, and the estimates become more precise, and estimation error becomes less of an issue, these polygenic scores will often have substantial predictive power. So we're at the cusp of remarkable scientific opportunities, but there are also important sort of ethical and policy challenges. My view is that there are many productive ways to use these polygenic scores, but a number of important interpretational caveats applied, and I think it's a good idea to put a lot of thought into interpreting what exactly these genetic predictors are, and not assume necessarily that they reflect something fixed and immutable. The mistake, I think, is to assume that because the SNPs, your genotype itself, is in a certain way immutable, that its effects have to be immutable, and of course that doesn't follow from the observation that your genotype is fixed. It doesn't mean that its effects are. So that concludes, and I'll hand over to Daniel, who's going to give us show us some applications of polygenic scores.
So this is a representation of a fragile family taken from the Princeton Alumni Weekly. They did a report on some of our work, and you see that while the mother is there and the children are, the father is coming and going, and that's the definition of our fragile families. Fragile Family Project was started in around 1998 by Sarah McClanahan, Garfinkel, and their colleagues, a group of developmental psychologists, sociologists, and economists. It's a probability sample of about 4,900 births, 20 cities, large cities across the United States, 75 hospitals, so they had to go to 75 different IRBs, if you can imagine. There were 3,700 non-marital births, about 1,200 marital births, in part serving as a control. And as you can imagine, there was a high prevalence of severe adversity and stress experiences. The cities are shown here. They're clustered over on the East Coast, but we do have Oakland, San Jose, and Austin. Because of the sampling method of this birth cohort, 47% of the children are black, 27% Hispanic, 21 white. The poverty ratio at birth was a median of 1.4, so these were distressed families. 35% of the mothers had less than a high school education, 30% had completed high school, 24% had some college, and 11% had a bachelor's degree. This image depicts the sampling strategy or the sampling history. There have been several surveys, beginning with the baseline survey, which consisted of a core survey of the mother while she was in the hospital giving birth or after birth. There was a detailed medical records extraction that was done by yet another economist, Nancy Reichman, when she was at Princeton, and a core father survey. The mother and father surveys were repeated at first year, and then a number of other surveys, including a primary caregiver survey, an in-home assessment of the child and the environment, a child care assessment in 10 of the cities, and again, repeating the father survey, was done at ages 3, age 5. Now there was a kindergarten survey at age 9, a child survey was added, and at that time I joined the project, and DNA was taken from each of the mothers and each of the child participants. We had about an 85% uptake to collect DNA. The DNA tests were repeated at the age 5 wave, which is the wave that just concluded, and RNA and fMRI studies were done in a subset of about 300 in the Ann Arbor, Chicago area through a separate grant. I assumed you'd get copies of these slides, so that's why I put this slide here. This is a summary of the kinds of data. It's actually a summary of a summary. But we have data in child health and development collected, as I just showed you, youth schooling and work, parenting, household and family, and parental health by survey. We have very little actual health data, although that's going to change at age 22, as I'll tell you at the end of my talk. Now, what I'm going to talk about now is the biological data and how we've begun to apply it. Some of this data is still in the final stages of collection and collation. We have genotype data, which I will make public probably by the end of this year, 2018. This is done using the psycharray, which is an instrument that has about 550,000 variants. It's a fairly well-distributed instrument in terms of the genome space that's used. And it can be imputed to 12 million variants with a high degree of probability, although there are some issues related to ethnicity that David touched on and that I'll touch on again. We believe that we are going to be funded momentarily to genotype the mothers, and we'll use a multi-ethnic array instead of the array we used. The multi-ethnic array has a structure that respects the genetic architecture of different ethnicities. And so we think that any polygenetic scores that are derived from this array will be an improvement, although they won't be perfect. We called copy number variants. These are areas of deletions or duplications in the genome, which I'll talk about in a minute. We have DNA methylation data at the 2 periods, ranging from 450,000 to 850,000 CPGs, which are the genetic locus of methylation. We'll make this public in 2019 and 2020. We have telomere data, and I'm going to show you some slides from a couple of papers. I did that because the original, my original description, which is changed in this booklet, that was sent to me, extensively dealt with my work about telomeres. And so I thought maybe there's a burning desire of the group to hear about telomeres, and so I will talk about it. We already have made public a list of about 10 or 15 candidate genes that have been studied, and we have this data, fMRA and cortisol, various stress tests. Okay, so some of the initial application of the work we did concerns the telomere. And the telomere, as you can see, this is a karyotype, so this is an image of 46 human chromosomes. This is what they look like in mitosis. They don't look like this most of the time, but when the cell's getting ready to divide, they look like this. And the chromosomes themselves are painted blue or stained blue, but the telomeres, the very tips of the chromosomes, about 8,000 base pairs long, so there are 96 of them, 92 of them, because there are 46 chromosomes, the telomeres have been painted yellow. Right? So you can see that the telomeres are present. And the reason that nature has engineered these telomeres, we think, is that with each successful cycle of reproduction of the DNA, when the DNA is duplicated, the end tips, the final tips, the edges of the chromosomes here, just because of the way that DNA polymerase works, become shorter by 50 to 100 base pairs. In other words, the enzymes that duplicate the DNA during reproduction of a cell are unable to duplicate the very tips of the chromosomes. Right? So that's basically the way all eukaryotes are designed. All animals are designed. Okay? And so the telomere, for those of you who are sailors or boaters as I am, serves as a sacrificial anode. Right? The telomere is about 8 kilobases long, and every time we reproduce a cell, right, about 50 to 100 base pairs comes off. And you might ask, well, what happens when it's all gone? And what happens when it's all gone is that cell lineage stops dividing. And if that cell lineage is a stem cell lineage, you get old. Okay, so one of the key drivers of human aging, or at least of the gradual failure of our systems, is telomere length attrition. It's a normal phenomenon. This is a picture of the telomere. I just wanted to, so this is the chromosome, this is the tip of the chromosome. It doesn't come out like that. I'm just sort of, the artist took liberties. This is the double helix, and at the very tip of the double helix, you have this complicated arrangement of proteins and DNA, and we'll leave it at that. Now, an interesting experiment of nature, and of course most advances in molecular genetics are experiments of nature. We find a patient with a disease, and then we try to understand it. This is a child with a disease called dyskeratosis congenita. Dyskeratosis congenita, dyskeratosis referring to failure of keratin-bearing tissues, so the nails don't form, the teeth don't form properly, the skin is flaky and subject to a lot of harm. They have a short lifespan, and the problem that they have genetically is that there's a mutation in one of these genes, and so their telomeres don't grow, right? Their telomeres are short, and so their stem cells fail very early in life, okay? It's sometimes thought of as a premature aging phenotype. Okay? Now, there are more subtle manifestations of telomere disease. So disorder like emphysema, right, can be caused by a mutation in another gene in this complex, right? And it's a mutation which shortens telomeres but doesn't shorten them as much as the dyskeratosis mutation does. All right? There are actually a whole bunch of human diseases that used to be thought to be diseases or disorders purely of aging or bad behavior like smoking. Pulmonary fibrosis is another one, which actually are genetically driven. As people get older, as I explained to you, the telomere length gradually decreases. Right? There's a big drop in childhood. This really looks like this. And it's been known for a number of years that under increased stress, increased allostatic load, to follow the psychologists, telomere length attrition is accelerated. Allostatic load, it's a term for the physiologic adaptations to chronic stress. Maybe high blood pressure or high blood pressure reactivity. It's not a good thing. Okay, it comes from the stress physiology literature. It's not from the medical literature, and it's not from the biological literature.
And is there an absolute, how comfortable are people that they've got stress now?
Well, again, I think that people are pretty comfortable with that. There's a large group at Rockefeller that's sort of led the way. There are ways of measuring stress by measuring cortisol, by measuring how the blood pressure responds to a stress test like a cold pressor test. So somebody who's had chronic stress or has a genetic predisposition who gets their hands shoved into near freezing water will have a higher blood pressure excursion and pulse excursion than people who have not. But it's not my area of expertise.
Is there a notion that some people are genetically, I'll get very, very loosely, more able to handle stress than others?
Yes.
Okay.
Yeah, so that, I mean, one way, one notion is Belsky's idea, not Dan Belsky, but his father's idea of the orchidandelion hypothesis, which I'm going to show you in about 2 slides. Okay, so that's it for telomeres. Telomeres get shorter in all of us. They're a normal part of getting older. In most people, telomeres are long enough to last till we're 50 and 60. But premature telomere shortening is seen under stress, as I'll show you in a minute. What that means biologically is open to debate. Is it just a biomarker, or is it actually a marker of a problem? Okay, so the original paper was by a psychologist, Alyssa Appel, who noticed that the chronicity of caregiving, how long a woman was taking care of a sick relative, basically, was associated. So this is chronicity of caregiving in years, and this is the telomere length of the woman, and she noticed this relationship. Okay? This has been reproduced over and over again. There's still a fair amount of debate, and I'll show you why in a minute, but I think it's pretty clear that there's consensus that telomere length shortening is a function of chronic stress. This is from Fragile Families, a paper I published a few years ago, and it shows that in mothers or their children, boys actually, we tried to use a very homogeneous population here to avoid genetic effects, who were advantaged. And here we're talking about not only advantaged in terms of how much income there is, but the presence of a father, the absence of maternal depression, the absence of harsh parenting. We had a 5 or 6 point sort of ACEs-like scale to do that. You see that both the mothers from disadvantaged households and their offspring have shorter telomeres. And these differences are highly significant and highly reproducible. So we see this in fragile families. The next study uses a far larger sample. It uses virtually the whole sample at age 9. And it looks at the issue of father loss, right? Can we measure the amount of stress these children are perceiving from father loss? Can we isolate that based on changes in telomere length? And secondly, is there a genetic component to this? And I'll show you that there is. So we looked at the reasons that the fathers were gone. Either they were divorced or separated or kicked out of the house by the woman. Either they were incarcerated, and given our population, you won't be surprised to know a lot of the men were in jail, or they died, and the same is true. There's a lot of violence around our families. And the first finding is that, in fact, and no surprise, loss of the father, any loss of the father before age 9 is associated with telomere length shortening. And here we've used all of the controls that you would think we would use: family income, style of parenting, race, gender, that sort of thing. Okay. And it withstands that. Now, the kind of loss actually seemed to matter. And these differences, I should have shown the p-values, but a sensitivity analysis shows these differences are significant. Separation had the least loss, incarceration a bit more, and death of the father the greatest loss. Right?
Sorry, those are changes in the length?
No, those are the actual length. So the length is shorter.
So you're not measuring the loss in telomere length.
Well,
It's just that's the difference.
yeah, so compared to no loss.
I see.
Okay?
So
We-
the
we
idea
would-
is continuous time. I mean, ideally, you'd really find out.
Well, we ideally, we might use a longitudinal assessment.
That's not this.
That's not this. And we do have 2 time points. But since most of this occurred before age 9, and our 2 points are 9 and 15, that wouldn't be helpful. So we're comparing the average to these particular subsets. Okay? Now, some of this is actually income mediated. Okay? So a mediation analysis showed that about 95% of telomere loss, when it was just due to the father leaving home, was due to economic changes in the family income. Less when the father was actually physically removed, couldn't visit, right? The children couldn't see him, right? And then it was 30 or 25%. Okay. Now, there's a gender effect. Oops, I went the wrong way. Sorry. Yeah, there's a gender effect. And it's the one you might have predicted if you thought about it. The boys seem to, this is a percent decrease, right? So again, it's not a decrease temporally. It's a decrease relative to the population average, right? So boys had a greater effect, right, than girls did, okay? And if you calculate what I call a serotonergic sensitizing score. So this is composed of 3 different SNPs in the biochemical physiologic pathway for handling serotonin. Okay, so it's the serotonin uptake pump. It's the enzymes that make serotonin, and serotonin has been repeatedly by us and others associated with environmental sensitivity. Okay? So these individuals, right, who had high sensitivity score, low sensitivity scores, right, did not have a major response to the father's departure. Those, on the other hand, who had the highly reactive serotonin alleles, right? The ones that have been repeatedly associated with heightened environmental sensitivity, in fact, had the greatest amount of telomere shortening.
This is a case where it looks almost like a candidate gene thing. Kind of
It is a candidate gene. It's a candidate gene. The reason this works, right, there's, I didn't want to get into this polemic about candidate genes, but candidate genes are perfectly fine to study as long as they have medium to large effect sizes, right? The problem with candidate genes, the way psychologists and social scientists, economists have used them in the past, is that they were, in my opinion, low effect size genes. So the false discovery rate, or the false positive rate, was very high.
That's the guy who pointed most of it out.
Am I right?
Yeah, sure, of course.
Yeah.
There are candidate genes with large effects, and then you're
Right.
well powered to study them.
Right, and that's what I think is going on here. But it's definitely a candidate gene. I picked them because I thought they would work. Okay, so this is an example of the orchid dandelion hypothesis, differential sensitivity. There are people who are dandelions. They're very robust, but they're boring, according to Belsky. And there are people who are orchids. They're beautiful and sensitive, but they need very closely controlled environmental signals. Okay, orchid dandelion. Okay, so that's it, I hope. Oh, no. Now, I don't want you to believe everything you read about telomeres, because many laboratories measure it very badly. And there's a lot of hokum in the literature that's really due to measurement error. So I did an experiment a while ago where we collected saliva, because that's how we get our DNA and a lot of other people do, and we followed the manufacturer's instructions, and we left the saliva in the collection kit for up to a year. Then when a year was all done, we had taken aliquots at different times. We took different kits, took the DNA out, and you see that if you leave it for a year, the telomeres get shorter, right? And this isn't going to mean much to you, but this is a gel, and the first side shows that all the DNA is up here, it's intact. In other words, we ran this on Agros. It's up here, and here, all the DNA is broken up into little pieces, and so it was able to migrate down the gel. So the DNA has been degraded, right? So it's very important to be suspicious of telomere studies. So now I'm going to turn to genetics. Okay? This is a human chromosome, a human karyotype, right? It's a male, sexually a male, right? And in this case, each chromosome has been painted with a probe that's specific for the chromosome, so each chromosome is a different color, right? And you see that there's two of each, as David was saying. Each one has an allele, right? For each gene. One is from the mother and one's from the father. Now, the striking thing about humans, and actually all other mammals, is that we're all very alike. Right? Notwithstanding all the SNPs that David spoke about, we're 99% alike genetically. Right? So all of the differences in humanity are encoded in less than 1% of the genome. In fact, the typical genome differs from RefSeq, the reference sequence for human genomes, by about 5 million sites, or 0.15%. Okay, so it's really small. It's really quite amazing. And that's in part because 99% of our DNA, or 98.5%, is non-coding. It's there for important reasons, but it doesn't specify a protein. And so that's my analysis of genetic inheritance. It's pretty funny, actually. Okay. So you've heard about SNPs. This is how we might measure them. This is actually a sequencing run. And it shows that at this position, there's a C in this person and a T in this person. Let's say the C were present in 10% of individuals in this room, then this would be the minor allele. And you could code all the people in this room just by specifying them as 0, 1, or 2 with respect to this minor allele. I'm going to skip copy number variance because I don't have enough time. It's another form of genetic difference. Okay. Now, again, 99% of variance between us are SNPs or indels, small deletions or not. These copy number variants, which are big deletions of big chunks of DNA that differentiate us, and we each have about 500 or 600. Even identical twins have are different because of copy number variants. They're actually the bigger source of a bigger source of genetic variability than the SNPs David and I have been talking about. And in fact, that's why one reason for the missing H squared, narrow sense H squared. In a population, most SNPs are rare, and that's because there are many singletons. There are many SNPs which just one or two people have. But in an individual, most SNPs are common, right? And only somewhere around 1 in 4% of an individual SNPs have a frequency less than 0.5%. Okay, so the SNPs that David's talking about are for the most part common. That is to say, they're present in lots of people, but in different combinations. Now, here's a chart that shows that the number and the distribution of SNPs differs with ethnicity, right? If you look at this as the number of variants per genome, right? These are just abbreviations for Japanese, for Central European, and then all of a sudden you see it jumps up in the area I've circled, and those are African populations. And consistent with the theory of the evolution of human beings from a sub-Saharan African nidus, genetic variability is twice as high, approximately, in Africans. And that's part of the reason for the problem David mentioned, and there are others. Okay, so you can't just do a GWAS on one of these groups if you want to capture Africans. You have to do lots of GWASs because they differ a lot. So back to fragile families. We genotyped 2,900 of the kids. Here's the ethnic breakdown. 603 were European, 1,400 African American, 748. We used some filters that I'm going to spare you.
How was that determined?
How was what determined?
You said this.
Sorry.
Given we're talking about genetics here, I just want to know it's just how was it determined that somebody was white?
I'm going to show you in a minute. You know, I anticipated that question.
Okay.
It's a great question. Okay, so that's what this slide does. So if you go to something called 1,000 Genomes, which is a government database, and they've sequenced actually people from many different parts of the world. And they have listed them not by race, but
What's the organization?
National Institute of Health, and many people all over the world working in collaboration. And you can download this data, that's what we did. And so you then, these are, okay, I think I can finish maybe. These are ethnic, not racial, okay? So these people are from Southeast Asia, right? Or it's not ethnic, it's ancestry.
Yeah, we don't even know what race means, so.
Right, so I'm going to show you what it means in the next slide, right here. What it means to us in fragile families. So these are European, Northern Europeans, the blue people over here, right? These are East Asians here. And so this is really an ancestry marker. Okay. Now, if you take the fragile families data and overlay it, what you see, and here I've designated the racial statements of the mothers.
Okay.
I'm black, I'm Hispanic, I'm white, okay? You see that the whites, right, are closer, right, to the Europeans. Well, that makes sense. There's quite a bit of spread though, right? So we have a fair amount of admixture in our population. The blacks, at least using the first and second principal components, occupy a lower portion of vector here, okay? The Hispanics are sort of a smear, right? Ranging from admixture to African to white, which makes sense also, right? So the fragile families, and the other point is that most of the fragile families mothers sort of get it right, mapping their race onto their ancestry. You see that? Okay, all right. So that's what I mean by it. Skip this slide, okay, skip that slide. Okay, so polygenetic scores could be helpful to us and to the people I work with because they allow us to stratify people genetically to try to extract the environmental signals. That's the real impetus that the fragile families got involved in this. It reduces the burden of multiple testing, so I don't have to test 100 different alleles and then have to look for a false discovery rate. I'm testing one number, right? And it's easily incorporated, as David said, into a complex modeling system. Maybe I'll skip that. Unfortunately, we have a problem in fragile families. This is my last 5 minutes. Okay. Most GWAS samples represent adult-oriented outcomes, right? Not pediatric or child outcomes. We don't think they're going to be very large samples of children for a number of years, so we're stuck with adult data. Ethnic diversity is very limited. Almost all of the large GWASs, the kind David put together in his great paper, 1 Million People, are biased heavily towards Europeans. Right? And that matters for genetic reasons. Right? So our solution is to harness adult-based studies. And now I'm going to show you whether it works or not. Okay, so these are the polygenic scores we used. We used one for Alzheimer's disease, one for general cognition, 2 for educational attainment, the OAKB, which is called EA2, and EA3, which is David's score. Just for those who are aficionados, we didn't have permission to use the 23andMe data that David did, so our EA3 is modified. This data is not there. Okay? Height and BMI. So I'll show you how this works in our kids. These are the things we measured. We measured the PPVT, which measures receptive vocabulary, digit span, auditory short memory, the Woodcock-Johnson passage comprehension test, which is reading comprehension, and then a math test, a Woodcock-Johnson math test, height and BMI. So let's see what we can predict. So here we're looking at the variance explained. These are European ancestry, I almost said patients, subjects. And this is the partial R square, which is, I think, similar to the term you were using. And so you see a couple of things. If you want to predict, using the PBVT, the EA2 score, we don't have EA3 at this time, predicts pretty well, maybe 4%. I mean, maybe the partial R score is about 4%. Interestingly, and this actually is something we've looked at again and again, a PGS developed for predicting Alzheimer's disease in older adults predicts digit span memory in children. Right? So this actually caught the attention of NIA, National Institute for Aging, who gave us a lot of money to measure ApoE4 in these subjects.
So what is digit span?
It's I'm going to tell you 4 numbers, or 5 numbers, or 6 numbers.
Oh, and how many do I remember?
Right.
And so you're saying that there's a correlation between that and later development and parental Alzheimer's? I just want to understand the result.
No, not parental. So we took a general GWAS. I'm going to sit down for a second because I hurt my knee. A general GWAS that predicted Alzheimer's disease, and that was a GWAS set up to find genes, and these people have Alzheimer's, these don't. You want to find the genes, okay? And then we took the effect sizes that they discovered and applied them to our children. And lo and behold, it picks up a memory test, right? And it doesn't pick up every test, it picks up a memory test. So we thought that was, pretty cool. Okay.
So I just wanted to clarify, so the training sample is adult training?
Adult people with and without Alzheimer's disease.
Yes, and then you use it to predict
In children.
It turned out, so okay, so does
So
that mean that?
Ken Langer has this kind of, very people in Michigan, I think, there is a life course. Alzheimer's is a very complicated thing, basically onset, age 0, or maybe fetal origin, and that is what they're playing with. Ken
Could be.
Langer says it's active. It's nothing to do with the various plaques that they find aren't to do with it. It's a life course.
So,
No, you couldn't take that, you couldn't draw that inference. I mean, this is
I'm not
significant.
drawing it.
No, but it's explaining 4% of the variation, right? So, yes, there may be a genetic component. If it explains 4% of the variation, that's something, but that doesn't mean that all the plaques and so on aren't.
no.
Who's drawing an implication of that? I'm saying that Ken Langer had a prior theory of that. He's been developed, and many people have been developing the life course vision of Alzheimer's. This is not inconsistent with it.
Mm-hmm.
Good.
So I don't want to give you my interpretation because I want to get through my slides. I'm about to get the hook. We see that EA2, educational attainment, also predicts about 8% of one's performance on the math test, and the height, right? The height PGS, when those Ks are extracted and used in our sample, picks up about 7% of the height. Right? It's not too bad considering we have a small sample, and these are kids, and that's adults. Interestingly, when you do it in African Americans, you see a much worse performance. And I believe that this is not an accident. This has to do with differences in the genetic architecture that are not really resolvable. If you can, this is just another chart that compares it. Here we're using the proportion explained, height PGS, whites and black, educational attainment, using EA2, whites and blacks, educational attainment of the mother's educational attainment. So we're looking at the kids and we're predicting how well educated the mothers were, because 50% of the kids' genes come from the mother. You also see a failure with the African Americans. Now, very interesting, and something that I thought about this while David was answering a question. I think you see there's a variation in how predictive they are with the child's age. So the educational attainment predicts 8% at age 3 and 9% at age 9. We don't know at age 15 yet. Conversely, if you look at height, a physical measurement, the predictability increases with age. Okay? If you look at BMI, you see the same effect, right? The partial R squared increases here in Europeans, right, as the children get older, right? Increases less because the whole effect is diluted in other ancestries. Okay, you got the point. I'll stop here, and you'll have to wait till next year or next century to learn about epigenetics from me.
All right, so,
Thank you.
all right, there we go. All right, so like I understand I have an hour or so to get through all of these slides, and so I'm going to relax and, so thank you, first of all, for having me here. I spend a lot of time worrying about data and thinking about how I can bring to bear new data in order to answer important questions about mostly, I think it was education issues, but increasingly issues more generally about child well-being. And I have not really thought about biological measurement, since I finished college. So, I did think about it in college, but this is not my thing. So you'll have to forgive me a little bit here. Okay. I want to talk first about an important application for this thing, and that is to think about special education. So when you think about biological issues that come up in education issues, one of them is this sort of thing. Is that we've seen this sort of striking growth in students with disabilities. It's increasing both in the number, percentage of the population. Kids in special education do worse than general education kids. This has been kind of persistent. They do worse in everything, in reading, in math, in 4th grade, in 8th grade. They are less likely to graduate from high school. But we've been working on this for a while, so the fact that things are actually, in a sense, not getting better, but holding steady is worth thinking about. And so this just gives you an idea of how this is across the cities of the United States. It's not one place, it's every place. And you could sort of pull these numbers off the internet. I did, that just sort of show you how we do see these, the up and down of it, and particularly, I think, in urban areas. So there were a whole bunch of, of course, for our policy geeks, and this costs a tremendous amount of money. So the amount of money that we spend on special education is not trivial, and it's not slowing much. And so one of our continuing issues to figure out this is, what's going on here? Is this about our changing educational systems? Is this about our changing definitions? Is this about changing identification? Is this changing underlying problems? It's sort of things that sit right at the nexus of health and education. And I think we don't really have good answers for it. Learning disabilities is one that that David brought up, and I think that's kind of a particularly important one because it's not something that we often find out about until a kid goes to school. So identification is a real issue. Common learning disabilities would include dyslexia. It's possibly the one you hear about most often. But, there's dysgraphia, which is dyscalculia, which is about similar sorts of things in mathematics. Dysgraphia has to do with writing. I think the view from the field is this is a lifelong challenge. This isn't something that is cured, or fixed, but it can be something that can be remediated in the sense of you sort of teach kids how to deal with it. So you teach a child to read, although they may have had trouble learning to read. Once they do learn to read, they do better. So, what's important about this is again, because it's not exactly clear where that comes from and when it happens, but most people think that early intervention matters. And so, just to give you another take on this, this is data from New York. In New York City, for example, our students with disabilities are substantially kids with learning disabilities. This is like a big share. So when you think about special education, it could be you're really thinking about these kids, the other health impairments, which includes, these sort of low incidence things. But the point is, this is, a lot of people in here, and a lot of them are in this category. So it'd be really helpful if we could get these kids early, and we don't, right? So this is just tracking a couple of cohorts from New York, and this is the overall classification rate of students with disabilities. But it's the students with learning disabilities who just sort of pick up over time. So if you see the idea, this is just following a group of kids through the grades. So in many cases of disabilities, kids arrive in kindergarten or in the year before something, and they're identified as having disabilities. This is speech impairment. Speech impediments, or in some ways they can be addressed. Some of the others do drop, but learning disabilities go straight up as they start to identify kids with learning disabilities. So real need to identify them. What would be another kind of example is childhood obesity, just going right up. Keep hoping it's going to stop, it's going to slow. These are not things that nobody's paying attention to, and yet we have some continuing issues. So in the case of learning disabilities, I think there's actually relatively little rigorous evidence on this. And part of the reason is that it's hard to get the requisite data. If you want to look at kids with learning disabilities, you typically want longitudinal data. You want information on their specific disabilities, their special education classifications. You need a sufficiently large sample, and the potential endogeneity disability classifications, which is where I think some of these things, these biological markers, could really come in handy. How do we think about these? How do we try to peel apart some of these things? Is the rise of ADHD just prior to the beginning of standardized testing in high school the response of anxious parents and children who want more time on tests? Or is there really something going on here? And is this something that we should have been identifying earlier? So, biological data of all kinds could really help to try to figure out what we should be doing and who are we missing. And so I want to say, this is, I could have chosen 5 different examples, but it seems to me special ed is an area where there's new data, that's both education data and child development data, and biological data could just be transformative. Good to know. Why do some students excel and other students lag under different circumstances? Why do some of them become overweight and obese or anorexic? All issues we see. What is driving the rise in autism? And is this something about the environment? Can we start to pick these things apart? These are all first order issues, I think. So these are, not small fringe issues. These are important ones. I would sort of put, I think this probably mirrors to some extent what you have in your presentation. There are 2 kind of first order ways to get at this. The first is to use these sort of data to drive identification. So if we think early identification of problems and potential issues is important, a great way to do this could be to use these sort of genetic data in some ways to figure out who's likely to be needing services. Maybe it means we're less likely to misclassify or underclassify or overclassify kids, but it means we're going to do better at identification and potentially do identification earlier, all of which would be great. It's also possible we could just do better with the interventions that we do. It might turn out that some kids, some circumstances, some types of underlying conditions will be more amenable to remediation in one way rather than another. For some kids, the underlying problems are going to be addressed by Wilson method reading, and for some kids, it's going to be some other thing. And it would be good to be able to target those better, which we are not so able to do now. I think a more complicated thing, but I would push the group to think some about, is that there are these a little bit more aggregate kind of questions that we always seem to be pushing off the table. One of the issues about special education is we've got these incentive issues in funding and in policy. It's not about an individual level thing. There are also population level questions that could be interesting and important to think about, and I'm probably going to come back to that. That would be helpful. Like, is this, if you could kind of come through with a genetic characterization of the propensity for a population in a particular area to have children with certain kinds of conditions, then, and compared to the next town or the next school district, you could, design appropriate policy so that they're not over-identifying kids, pushing kids into special ed who shouldn't be there, or vice versa, or underfunding it. So in my way of thinking about this, we end up with two different kinds of data. And I'm sure this is way too crude, but like to me, there's the time invariant kind and there's the time varying kind. I think they suggest different things we might do with them. First thing I want to ask on these time varying ones is like, are these something that we're ultimately going to be able to do to use as outcomes for things? Is this what I should be thinking 25 years from now when I'm not doing this anymore, but somebody else is doing it in my stead? Are they going to be, tracking the telomere length to see what the different things do on stress levels of kids? I understand cortisol studies are already underway in various places. Maybe I'm wrong. But that would be really useful to know something more about. Are these ultimately going to be helpful to capture aspects of kids' responses to things as outcomes, as compared to just as control variables? I promised I was going to ask you about this. I see on a slide it says genetic data can be thought of as randomly assigned, conditional on parents. I mean, what is not to like, right? I want something that's randomly assigned all the time. But in the end, I sort of kept nagging at me. Does maternal or paternal health at the time of conception shape which eggs are fertilized, which ones result in live births? Nice way to how to differential pregnancy termination affect what we see. So, if, for example, some combination of things ends up in a spontaneous abortion because it's problematic for the baby, then what does, how does that shape the sample of what we see in the births? Does it matter that parents aren't randomly assigned? And how do I think about that? We think lots of evidence that there's some kinds of assortative mating. I don't know. So I don't know that any of these things are a problem, but I know that it leads me to try to think about things. Both of these studies I see are about associations. So when I get up in the morning, honestly, what I'm thinking about all the time is things like, what are the causal effects here, and how do we exploit them to make the world a better place? And what I really want to know is not, do kids who have lost their fathers have shorter telomere lengths? What I really want to know is, how does father loss does that lead to shorter telomeres, and should I be worried? And I don't think we're there yet. And I don't know how we get there, but I think we got to get to those questions. Another example, why do school interventions improve weight outcomes only for those with high BMI PGS, if I got that right? And should we be doing something about this, or should we not? And what should people be doing? These raise a bunch of issues about what's the scope for intervention, and both what would the consequences be? Now, every time I read one of these things about the relationship with blah, blah, blah with attainment, I wanted to send you this slide. The thing is that school context is going to matter a great deal. It turns out attainment is a pretty crude measure of educational outcomes. It's true people use it all the time, but it's, I think, nobody's happy about that. And it's widely varying across jurisdictions and time. What does it take to graduate from high school? What does it take to graduate from high school in Alabama today? And how different is that than what it takes to graduate from high school in New York? Now, come back and tell me it's the same thing, but I don't think so. What does it take to graduate from high school in other countries? There's big variation, not to mention high school to high school within the same place. And so if we're going to look at, graduation as an outcome, and we're going to use that to back out scores or something, I think we got to think a bit more about the geography of this. And so, in my other life, I do work on urban economic issues, and that leads me to worry a lot about residential location and where do you live and how do I think about that. And so imagine your genetic composition, say, is correlated in some way with your ethnicity or your race. And say we live in a world in which there's residential segregation around race or ethnicity, which I think we do. And say it's also the case that those things are correlated with the quality of the schools. Say certain minorities tend to go to schools in low-income places with poor school outcomes and poor schools, then it's hard to figure out, what do I make of this? Am I sort of pulling out something here which is about people of color live in bad neighborhoods and have bad schools because they're poor, or, so I don't know quite what to do with this, because what I wanted to do is say we need much better spatial econometrics in this. We need better data. And then I look at the N's and I think, the N isn't big enough, not in fragile families anyway. But it seems to me we got to think some about the spatial aspect of this. I'm a huge fan of linking to administrative data. I want to say there's a whole bunch of other things you could do. It'd be really interesting to look at just validating data. To what extent is what we have that identifies kids with things that we think have genetic components, do we see that in the same data sets? Are they, how good are they? And are there kids we're missing? So really, really super interesting. Obviously there's heterogeneity and treatment effects. Love this idea of controlling for these time varying stuff about stress. So as an example, I did some work a couple of years ago about the consequences of residential mobility for kids' academic performance. That's residential mobility, school mobility. It is bad for kids. They do worse. One possibility is that it's stress. I could tell you 5 other possibilities, and we're trying to tick them off. But it would be super cool to be able to do something like this cortisol thing or this telomere thing, right? To be able to say something about, oh, no, this is stress. And obviously, the direct thing. We have really few population level measures of health consequences of educational interventions for children. Now that said, sorry, I would have been remiss. Somebody's got to be the party pooper here, right? Like I'm not volunteering for my kid to be in this, right? Are you? You doing these? You want your who owns that data? I know these are really tough issues. I don't have answers. What do we do then about using the potential data for making decisions about eligibility for programs, special education, affirmative action, yeah. And I fear that the public conversation tends not to be particularly nuanced about these things. That the study comes out and it shows, I see the correlation between association between blah, blah, blah. It's in, you know, the newspaper. And I don't know how you manage it. I don't know, because I don't see us having a great public conversation about this in comparison to all the other things we're having a great conversation about. Let me just say something about these administrative issues. I know, I only have 5 minutes, right? That's the luck? Okay. So I'm just going to leave you wanting more. But the first thing I want to just say is part of our problem is the FERPA-HIPAA, you know, argument. Who's, you know, what's the prevailing code there? And I think there are ways around this, but one of our issues is the federal government isn't leading. And I have said this, you know, every time I get a chance. Maybe this group could say it louder, but that the federal government needs to issue some guidance to states that tells them these are the rules. Because I think states don't know, and the folks in the legal offices, I think they're doing their best. They're doing their best, and they're nervous, and they have a right to be nervous, but that would be a good thing. One of the other things we need to think about is the difference in the underlying logic under which you're allowed to use data. I think HIPAA, it's much more around surveillance. And in FERPA, it's much more about educational outcomes, but we could talk about that. I'm going to just say a little bit about what happens when you can overcome these things, because I have over the last, long time, put together some really fantastic data around for New York City. I'm going to go through some. I can tell you about some more. There's more coming. This is something we did like 6 months ago. And I thought like, oh, I should show you something. So we have data we call the analytic files. This has all this stuff at the individual level. It's like the stuff you think it is, you know, it's whatever, test scores, anything you've ever seen in anybody's paper, you know, test scores, attendance, 3rd grade through 8th grade, blah, blah, blah. Got a bunch of data on the built environment that we've put together around New York City. New York City is large. We have over 1 million kids every year, you know, 1,500, 1,800 schools every year. So it's big, big system. We have data on crime, on the food environment. All of these are things that we've gotten from various sources to capture the neighborhoods around kids. We have a bunch of data on their housing, which you can get again from public and government sources. New data on pupil transportation, and I can say some more about that. It's how do kids get to school, or how do we help them? We have a bunch of data we call the restricted data. We have the fitnessgram data in which they take every kid's height and weight every year, and then they go and they do performance on, I don't know, like the sit and reach and the push and something. They survey kids about how do they feel about school, what are their impressions. This is every kid in grades 6 to 12. So, you know, tens of thousands of children every single year. We have data from the Office of School Food and participation in school meals. So every time they eat breakfast or they eat lunch at school, I know whether they do and what time they do. Most of these things roll out, meaning it's first in a few hundred schools and eventually it spreads to everybody. We call this special use data. This was designed to tell my grad students where the data lives. On daily data on absenteeism, this point of service, that's the school food, that's daily data on what you do. There's just all sorts of things. I mean, we know where the kids live, where they go to school. We know whether they participated in summer jobs programs. We know how they performed on the New York State English languages. Oh, no, maybe we only got that through 2015 and then they renamed it. So, you know, it's all sorts of things that mostly came from different projects. Every time we start a new project, we have to figure out what's the data we need to do it, and we go and we try to figure out how to get it. And we've mostly been pretty successful getting what we need. This is also the student level data I've been telling you about, but, you know, there's a bunch of things that are not, because some of this is not student level. It's about the neighborhood. It's about where do you live, and, you know, what's your block like, your facilities, where are the parks. I don't know, what do they do? There's crime data on violent and disruptive incident reporting. Homicides, assaults, property crime, violent crime at the block level, blah, 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 blah. Yeah, this is just more detail. This is what comes at the school level. This isn't everything, because as I said, this was, we were cooking it up to tell the graduate students what was in the stuff, and I thought this would be interesting. And we're, talking about getting more health data. So I think there are really, there's a gold mine out there. It's hard to work in that mine. But I think it would be even richer if we could add in some of these other things that would really help us get at some of our causal issues, like, are we creating problems for ourselves? Are these naturally occurring and we're solving? And how do we think about that? Anyway, I know I'm standing between you and drinks. So thank you.